And he's going to talk about bridging the gap between nanospray and clinical analysis, new approaches for automated proteomics. Nathan, welcome.
Thank you, Tim, and thank you to the organizers of Pittcon. It's a pleasure to be here. Can everybody hear me okay? Great. Okay, so a little bit about me first. I'm an analytical chemist by training. I actually had the pleasure of doing my PhD with Rick Yost and spent most of my time working with instruments called quadrupole ion traps. And this was back in the late 80s to early 90s. And the quadrupole ion trap at that point was an instrument that was new on the scene. It had tons of potential in terms of being able to do sensitive MSMS and rapid computer-controlled analyses. But the instrument that I found in the laboratory at Florida was actually not much more than a breadboard system. And so I really was incredibly fortunate to be involved in that in the early days and see it move from a device that was hard to use, didn't have a lot of practical applications, and then moved into really mainstream proteomics. It's one of the go-to instruments for the entire field. So I've been passionate about mass spectrometry and have enjoyed seeing technologies that are not quite ready for prime time make that transition. And I think we're now seeing that in clinical analysis, particularly with the MALDI analysis we're going to hear about later, that transition is incredibly exciting. So what I'm going to talk to you about today is my current field, which is protein analysis or proteomics, and particularly some of the challenges and technologies that I'm seeing and my lab's working on to move those to more quick, more cost-efficient, and more sensitive and selective measurements. So mass spectrometry is clearly an enabling technology for the clinical laboratory. It measures analytes with absolute molecular specificity. I mean, that's the key driver from my point of view. And it increases the accuracy of diagnostic tests, ultimately improving patient care. If we can make more accurate measurements, and we're actually measuring the molecule that was in the patient's body, that's got to be better than looking at radiometric assays where you're not really measuring the molecule, you're just counting or looking at a fluorescent output. Proteomics offers tremendous opportunity for the development of new assays. We can measure thousands of proteins by proteomics. There's many more thousands that we can't measure, but we can measure a lot of proteins. However, there's numerous challenges, as Rick highlighted some of them, that currently limit the applications of these tests. There are not that many protein mass spectrometry-based assays currently in the clinical laboratory. There's development of ApoA1, ApoB assays, and also thyroid globulin, but not too many more than that. I had the real pleasure of visiting Colin Creaser at Quest Diagnostics Lab in California, where about 4 years ago he walked me through their mass spectrometry lab, which was filled with 30 or 40 triple quads, all set up running 24 hours a day, 7 days a week, 50, 60,000 LC-MS measurements per day. And he said, really, it's incredibly simple. He was doing 1 and 2 minute separations for analytes like vitamin D, and it really opened my eyes to what mass spectrometry, the potential is for the clinical laboratory. The types of experiments that I do, we are working on 10 samples a day or maybe 25 samples a week. Those are big experiments. And so there's lots of room for us to move the technology forward. Key points I want to talk about today are some of the challenges for the quantification of biologically relevant proteins in plasma, incredibly difficult. I'll give you 2 examples of ultra-low trace analysis assays where we're measuring analytes at picomolar levels, so picomole per liter in plasma. It's not easy to do. And really, the focus of my talk today is thinking about how to make nanoelectrospray easy. We depend on nanospray to get the sensitivity that we need to measure many proteins. And making that something that's easy to do and readily accessible to the clinical laboratory is one of the major hurdles. And to do that, I'll talk about one of our really contraptions we've come up with, which is a multiplexing system for nanoflow chromatography that we think can be used to improve throughput. So Lee Anderson wrote this article in 2002 in Molecular Cell and highlighted basically the reference intervals, which is the height of those bars, for 70 protein analytes in plasma. And what you'll notice on the left is here we have the concentration range of those molecules in plasma. Again, the normal ranges are shown with the bars. And then you see that they span anywhere from 10 to 10 times 10 to the 11th picograms per mil in concentration. These are the classical proteins, tissue leakage proteins, and as we get down to lower and lower amounts of interleukins, all things we would like to be able to measure. 11 orders of magnitude is a big number, and if you think about it in other terms, the diameter of Neptune is 5 times 10 to the 7th meters in width, and compare that to a grain of sand, and now you're looking at or thinking about the 10 to the 11th order of magnitude. A huge number. How would we come up with an analytical method that could find a single piece of sand on a planet? I mean, that's really, if we were trying to cover the whole proteome, that's what we're looking at. We're not going to do it, okay? But just to give you a sense, the complexity of what we can see now, there's tons of room for us to be able to go lower. Now, for small molecule bioanalysis, the task is to find a small chemical marker in a complex mixture. It's a needle in a haystack. And fortunately, we have a pretty good magnet for doing this. It's LC-MSMS, and we're often working with a pretty generic prep just to get rid of many of the things that we're not, molecules that we're not interested in, so things like proteins and lipids. So a generic, non-specific liquid-liquid extraction or solid phase extraction is normally what's used. We need to introduce the samples into, of course, a triple quadrupole mass spectrometer, and so we need a fast LC sample interface, often using short columns, where we're really not trying to get much chromatographic separation. And we're using, instead of sometimes gradient elution, but normally isocratic. And then, of course, our really powerful magnet is the triple quadrupole mass spectrometer that can be tuned to look at a specific fragmentation pathway of a structure. So again, we're measuring the molecule that was in the body. For proteins, it's not so simple. Instead of trying to find a needle in a haystack, it's a piece of hay in the haystack. We're trying to identify one particular protein amongst a sea of 10 to the 11th orders of magnitude in concentration of other proteins. And so you're going to need more than a magnet. So one of the technologies is shown here. It looks very similar to the last slide, but it's actually quite different. We really depend for many, many analytes on specific preparation, where we're using antibodies to not necessarily give us the selectivity, but to enrich for that molecule. We cannot amplify proteins, or we can't amplify molecules by any other means. So we need to start with a larger amount of sample, often a mil or more of blood or plasma, and use immunodepletion or immunoaffinity to enrich for the molecules of interest. We do use chromatography. In fact, I'm going to talk about this some more in the next few slides. It's nanoelectrospray to ionize these peptides or proteins. And we have very, very complex mixtures, so doing a quick 1-minute separation is not an option for us. These are often as long as 2-hour separations to separate these complex mixtures. And again, the triple quadrupole is one of the primary instruments that we're using. Once we've figured out what proteins and peptides we want to measure, we'll use the selectivity of this device to hone in on the analytes of interest. So we get in sensitivity by enriching for molecules of interest using antibodies, and then selectivity by using MSMS on the triple quadrupole. So prior to coming to the University of Pittsburgh, I had the pleasure of being at Merck & Company for running a proteomics group there, and we were asked to develop assays for preclinical and clinical development for a peptide called glucagon-like 1 peptide. Okay, this is a peptide hormone. It's anti-hyperglycemic incretin hormone that is selected for endocrine cells and located in the intestine mucosa in response to the presence of nutrients in the gut. And what Merck was working on was DPP-4 inhibitors that prevent the conversion of GLP-1 from the active form, which is 30 amino acid residues, to the inactive form, where you cleave off the 2 N-terminal amino acids, histidine and alanine. And so all the immunoassays that we had for GLP-1 really were not sufficient for measuring the active and the inactive form of GLP-1. And so there was a need for being able to measure both forms. And of course, mass spectrometry, losing histidine and alanine is an enormous change in mass, so there's no difficulty whatsoever differentiating those 2 molecular forms. And so the strategy that was taken, is shown on the right-hand side here, is that we used a anti-GLP-1 antibody and linked that to tosyl activated beads. These are the dyna beads that are magnetic beads. and then incubate that with your sample. In this case, we were working with about 1 ml of plasma. The GLP-1 would be attached to the antibodies. You'd use a magnet on the side of the tube or on the side of the 96-well plate to pull those antibodies away from everything else. Rinse and wash, and then now you've converted, again, about 1 ml of plasma into a 30 microliter eluent off of the magnetic beads. You can take that straight into the triple quadruple mass spectrometer, where again you're going to have a pretty complex mixture that it gets ionized. Select your precursor ion in the first quadrupole, dissociate it in the collision cell, and select your product ion in the third quadrupole. And this is the type of data that you get. So here you'll see in the bottom 4 select ion chromatograms, where you're looking at first the active form of GLP-1 and its internal standard, heavy label internal standard, and the inactive form 9 through 36 of GLP-1. Again, keep in mind the numbers here. We're looking at single-digit picomolar concentrations. And just these types of instruments alone, they can detect peptides and proteins in the attomole range at best, but often what we're working with is femtomole amounts of material going on a column. So to get femtomole amounts of GLP-1, you really have to start with 1 or 2 mils of plasma and not lose any more than 50% of those peptides carried through this whole procedure. So it's really quite a feat to think about, this type of sensitivity. Okay, here's just some analytical figures of merit. These are the calibration curves for both the active and inactive forms, and the assay had a range of going from half a picomole, 0.5 picomolar, up to several hundred picomolar, and typically. Okay, just a little bit of LC-MS method. We're using a Xevo TQS triple quadruple mass spectrometer that was fitted with a trisaic nano source tile, so a manufactured nanospray column. Inside that column is C18. It's about an 85 micron ID and 100 millimeters in length and 1.8 microliter particle size. Just to give a sense of the sample analysis rate, this is we typically load this trap column at about 8 microliters per minute, and then use a gradient that's about 15 minutes long to elute GLP-1 into the mass spectrometer at 1 microliter a minute. So cycle time is about 4 samples per hour. Think about that for the clinical assay. That's not too bad. 1 sample every 15 minutes. It's nowhere near vitamin D, where it's 1 sample every 2 minutes, but it's not terribly long. But again, the clinical lab is an environment where they're thinking about quality control standards and calibration probably about every hour. So an assay that takes 15 minutes to run, plus adding in your QC standards, gets to be pretty burdensome. Here's some examples of the different QC samples. So you see a good signal to noise at essentially half a 0.5 picomolar concentration. And this is a test that was done in humans, where they did a meal tolerance test for 15 minutes and draw blood over the next few hours. And you can see both the active and inactive form of GLP-1 being monitored. Again, we're at the 4 to 5 picomolar level. So this can't be done by immunoassay, and these are the types of things that I think if you can make them easy enough, they'll become much more beneficial tests for the clinical environment. So another example, just to give you one more, is looking at stromal cell-derived factor 1 alpha, SDF1A. And we used very much the same procedure, where we're using antibody enrichment, using the dynabeads, the activated dynabeads. We avoid any kind of complicated reduction to alkylation, so it's a pretty simple preparation, 1-step preparation. This is being done on a triple quadruple mass spectrometer. And again, we're doing nano electrospray using a biobasic C8 column that has an inner diameter of 75 microns and is 50 millimeters long. These are more traditional pulled columns that have been used for proteomics. Cycle times, again, is about 15 minutes per sample, and we're loading at a flow rate of 2 microliters a minute and eluting at 1 microliter a minute. And this gives you a sense of the signal to noise. SDF1 alpha is not single-digit picomolar. It's closer to single-digit nanomolar. But the assay actually had an analytical range that went down to 20 picomolar, so quite a long linear range. Okay, as Rick highlighted, all these classical proteomics assays are not straightforward. They're not as simple as an RA, and that's really where we'd like to see them go. Owning a mass spectrometer takes a certain level of expertise and familiarity. performing all of these steps that are common in proteomics labs are not things that clinical techs are going to be familiar to do. And so again, the challenge is, how do we take these complex assays and make them more amenable to others? So the two assays that I just described are intact peptide assays. We're avoiding digestion, calibration. Internal standards are pretty easy. We're putting in isotopically labeled peptides of the same structure. And we can do quality control samples and interlaboratory comparisons. But the key thing is plasma is a bear to digest. Anybody that's done this, you're using an excess of trypsin, and if you have anything like heated up, you end up with a pot of glue. It's just not a good sample to work with. And so the sample preparation for proteomics is complicated, and then the apparatus is very complicated compared to what is typically found mass spectrometers are becoming more prevalent, but they're still very new. And nano-LC systems are very rare. I mean, in clinical labs, they're just a pain. Having run several different laboratories, both in industry and now academics, I would say one of the biggest Achilles heel for proteomics is the nano-LC systems. And so making them reliable, making them easy to use, easy to troubleshoot, actually sounds simple, but it's one of the major things that we face every single day. So peptide and protein quantification places great emphasis on nanoflow chromatography. We can't go, in most cases, to the flow injection type analyses. And why is that? Why do we use nanospray with small bore LCs? This is, again, an illustration that was provided by Gary Vlaskevic showing just a red dye diluted into the volume of solvent that you would expect to elute a compound off of a 50 micron ID column and a 4.6 millimeter column. So clearly, the dye is more concentrated here, and that's important. Electrospray is a concentration-sensitive ionization method. So the more concentrated your sample is, the higher the response you're going to get per number of molecules of analyte. Okay? So in practice, you want to be able to elute your sample in the smallest practical volume to get the highest signal to noise. If you look at what this comes out to in theory, here we're looking at different size columns, 4.6 millimeter, 2 millimeter, down to a 75 micron column, and the relevant concentration of the band for a fixed amount of analyte. And so what you'll see as you go from a 2.1 millimeter column to a 75 micron nano column, you're going to expect somewhere in the order of 150 to 200 fold advantage over that 1 millimeter column. So big increase in concentration. In practice, you don't see that type of improvement in signal to noise, and that's because the matrix is also concentrated, and so you have an interference between the matrix and the analyte. But there's been studies that have been done that showed a practical improvement of 10-fold increase in concentration or increase in signal to noise. So my laboratory's been using the Nanospray packed column emitters. They're shown here. It's just a piece of fused silica capillary that has been pulled to a precise tip and packed first with a frit, and then the packing is filled in with any other type of normal stationary phase. We're often using C18 for peptides or C8 or C4 for proteins. Shown down here on the left is actually a photograph of the electrospray ionization plume. So you'll see the tip of the electrospray tip where we have a voltage applied. That voltage makes a Taylor cone and then a jet, and then as the droplets start to form, they fan out due to coulombic repulsion. And this again is a blow-up of that nanospray tip where you can see the frit and the packing material. So an advantage of this design over other types of columns is that there's really no post-column volume, and so we don't see any band broadening, which again is important because it helps us achieve tighter and sharper chromatographic peaks, which improve sensitivity for low abundance analytes. So we have good sensitivity because of nanospray, but do we really need chromatography? Do we need to run these gradients? Because they're very, very slow and time consuming. There's been several papers, in this case highlighting that if you use just selected reaction monitoring alone, and I think Rick's slide showed it as well, you're often going to see multiple peaks just because of the really complex nature of plasma. In this case, this is looking at peptides from CRP and albumin in depleted human plasma. And so you'll see it's the peak that we are interested in was highlighted with a star, but there's other things that you'd have to separate away. If you use a 1-minute gradient or a 5-minute gradient, that's it's not going to be sufficient. And so high-quality LC is critical for protein quantitation. And we just noted selective reaction monitoring, or MRM, is not sufficient. We also need to be able to have good peak shape for quantitation to get high assay precision and accuracy. And as Rick also highlighted, proteomics or mass spectrometry, by its very nature, is a multiple analyte assay. So we have the ability in one analysis, or one 15-minute analysis on the mass spectrometer, to look at more than one compound, and so we want to be able to use that benefit. But when you start thinking about multiplexing assays, like the two I showed, you're going to have to introduce additional chromatography to separate out the proteins that you want to look at per assay, the different peptides per protein, and in some cases, well, the MRM transitions should all fall within the same time range. Okay. So this slide sort of summarizes some of the key requirements for Nanospray. You need a gradient pump. Often the pump is designed to run at flow rates of 100 nanoliters per minute up to 1 microliter a minute. And sometimes, because loading samples is time consuming, you'd have an additional channel to load samples onto your trap column more quickly. Here's the flow rates that we're working in, I just described. And the injection volumes are typically 1 to 5 microliters. So just a standard configuration, a source of solvent is your pump. We use lots of filters to prevent particulates from contaminating the system. Sometimes flow splitters, if we're not using a dedicated nanopump, we'll split the flow from a high flow regime down to a nanoflow regime. Micro injection valves, tubing, and then your Nanospray column or emitter into the mass spectrometer. Again, some of the technical difficulties that we encounter is due to the types of tubing, which is often fused silica, and the size. This is a blow-up of a fused silica capillary in a human hair, so this is 75 microns in ID. And the smallest things, even contaminants, particularly coming from water, carbon particles, tend to block these fittings. So how can we bridge this gap? How can we make Nanospray something that's more accessible to the clinical laboratory? We were very fortunate to work with a company called New Objective. I've had a long-standing collaboration with them. And what we really wanted to do, at least for my lab in Pittsburgh, is take all of these delicate parts that work very, very well and put them in a plastic casing that makes it modular and easy for people to use. So in my laboratory, it's an open laboratory. We have students coming in, investigators that are not mass spectrometrists. They can bring their column, drop it on the mass spectrometer, and attach their column to the LC system and be up and running in as little as 5 minutes. And so having something that doesn't require a lot of watchmaker skill to put on and make work is a huge benefit for us. And so we've been working with New Objective to think about ways to do this. And inside this plastic is the picofrit emitter that I showed on the previous slide. In this case, it's a 10-centimeter column and a metal union, and then just some fused silica transfer line that then connects to your LC system. So when you drop this on the source, it automatically makes an electrical connection. You can push this little button on the back, and the column comes out the front, where it's out of its sort of protective casing. We get very high performance nano-LC separation. So this is a microglia digest, and as you can see, we're running about a 90-minute analysis and seeing lots of samples, lots of peptides and proteins coming out over time. It's a pretty complicated mixture. And I just want to highlight is that often we're waiting, in this case it's not too bad, 20 minutes up front before we see the first peaks come out. This is just the time it takes for the gradient to get through all the plumbing in our nano-LC system, get to the column, and start to elute peptides off those columns. And then we have time after the run where we have to re-equilibrate the column for the next experiment. This is pretty typical. Sometimes, depending on what type of sample it is, you could have a 3-hour run where 1.5 hours is spent just either loading the sample or starting the gradient or waiting for it to equilibrate. Now, in the clinical setting, they would never tolerate this. In fact, I think one of the reasons why mass spectrometry has been adapted is because of the development of these multiplexed HPLC systems. So this is essentially 4 separate HPLC channels, 4 pumps that are connected to 4 individual columns, and then connected to a switching valve that cycles between the first pump column system to the second one, to the third one, to the fourth one, and to the detector. And so if you have an LC analysis that's 4 minutes long, and the peaks of interest that are eluting come out in 2 minutes, you essentially would start one system, make an injection, start the next system 2 minutes later, start the next system 2 minutes later, and start the fourth system 2 minutes later, and they would all just come out over time, and the mass spectrometer would switch to each channel and sample at the right time. This is what I observed running at Quest when I was there in 2008, 2009, and again, you saw a room full of 30 mass spectrometers, and since there's 4 HPLCs for each one, it was 120 HPLC systems. They're really important for getting the throughput that they wanted. Now, Dick Smith's group at Pacific Northwest National Labs had a really nice paper in 2008 with the same concept. They had multiple pumping subsystems, so multiple HPLC pumps, lots of switching valves with the same principle. Maybe we can time slice or segment nano-LC. Now, as you'll notice, there's a lot of plumbing here, and as I mentioned earlier, we're running at a few hundred nanoliters per minute. So pushing peptides and proteins through a system like this is going to take some time. Plus, if you had any leak here, it would be almost impossible to find, in my opinion. It's hard to troubleshoot nano-LC with a single pump system. Doing it on something with 4 pumps, and I counted, I think there's 56 components in here, I don't know how many connections, was more troublesome than I wanted to undertake at Pitt. They did a great job, it gave beautiful, here's the chromatograms they observed on 4 different columns, but you'll notice the retention times are actually relative. That's because it's hard to get 2 pumps to give you exactly the same gradient, and so they would have to align chromatograms based on the peaks because the retention times weren't exactly the same. So these were things we wanted to get around, and working again with New Objective and the team up there with Gary Blaskovic, we came up with this idea. It's putting 3 columns on a stage that moves in front of the mass spectrometer, much like a harmonica, so back and forth. And then we tried to come up with an innovative way to reduce the number of connections. And so working with Valco, who makes these micro selection valves, we came up with a novel design that, again, it looks pretty simple. You've got your 6-port valve, and then you have a rotor. But instead of the slots on the rotor going around the circumference of the rotor, they cross it. And that gives you some pretty interesting capabilities. And so I'll show how this works on the next slide. So here's what we have. We have a typical proteomics gradient. In this case, it's about 30 minutes long. There's only about 10 minutes of peptide dilution time in the middle of the gradient. This is when we're loading the sample, and this is when we're equilibrating. And so the idea is to have one pump that loads a sample in one column, while another pump elutes peptides off another second column, and a third pump washes a third column. So making those connections easy is with the 6-port valve. You have 3 input connections, so each of the 3 pumps connects to 3 inlets on the valve, and you have each of the 3 columns connect to the 3 outputs. That's all you need. There's no other connections or fittings. So it's really a nano-LC system that has 3 columns that only has 6 connections. And as you'll see in the current on this slide, I'm going to cycle through this in a minute, column A is right in front of the mass spectrometer, and it's connected to a nanopump that's running a gradient. 15 minutes later, you'll see that the pico slide moves to put the column B in front of the mass spectrometer. And now you have that column that was being loaded by pump A, eluting peptides into the system for analysis. Meanwhile, the first column, column A, is now connected to the wash pump, and the column C is connected to the load pump, getting ready for the next analysis. And so just by toggling back and forth, turning this valve 120 degrees, you can make this fluid connection without lots of T's or multiple valves or anything like that. So it's a very simple system. Here gives you an example of the data. We actually get very, very good reproducibility column to column, and pretty good reproducibility between the different columns. We're limited by the ability to get 2 nano columns that give you exactly the same separation. Every column has its slightly different characteristics, but we now have an opportunity to optimize that and make columns with incredibly high tolerances. So I mentioned before, it's a simple system to set up. It's got 6 connections. We get about a 3-fold increase in throughput. And so your assay, if you have a 3-hour assay for complex proteomics mixture, you can now get 3 samples run in that same 3-hour period. Channel-to-channel variation is eliminated, and we have a single pump that drives every single gradient. So any variation we see here is not due to a difference in the pump or different flow path. It's all the same for every single column. And I mentioned before that we have a pretty long time to load samples and a long time to wash the columns. That's also a huge advantage. Being able to go to small particles gives us better chromatographic separation, and having 30 minutes or more time to load larger volumes of sample directly onto the column, we don't pay any penalty for that because that's happening while the mass spectrometer is analyzing another column. So we can load very slowly and over large volumes without traps and get all the material on the head of that column. Similarly, carryover is an enormous problem in proteomics. Most laboratories will run a sample for 3 hours and then run another sample that's just a blank, just to wash things off their column. We have the full length of whatever our analysis time is to run multiple gradients over that column just to wash it off. So we don't have to run blanks in between. And I didn't mention it up here, but another advantage of this design is that there's always pressure applied to the columns. So in a normal HPL system, when you switch the injection valve, there's usually a pressure drop that occurs and then a repressurization step. The way this valve works is every single channel is a linear flow path, and so you always have pressure being applied to those columns. It makes it very simple to operate and to troubleshoot. So I'll just summarize here that hopefully we're now seeing in the session that mass spectrometry is enabling technology, and I think proteomics has a little ways to go, which is exciting to me. I think there's lots of problems to solve and places to improve it. Right now, I think most proteomics assays are simply too complex for the clinical lab. We're not there yet. Maybe 10 years from now, it'll be commonplace, but it's not there today. Affinity capture of undigested intact analytes is certainly achievable. I showed you 2 assays, GLP-1 and SDF1 alpha, where I think with a little bit of work, those can be adapted quite readily to the clinic. Nanospray chromatography, we tried for GLP-1 and SDF1 alpha to use normal analytical scale chromatography. We could not get the signal to noise that we needed. It's still essential for everything that we want to look at. And really the benefit, there's 2 examples, the trisaic columns from Waters and those pico-chips column from New Objective really gives you that reproducibility column to column that many proteomics labs that are still pulling their own columns don't get. So I think as we look at how to make things more modular and more reproducibly, the technologies will become easier to use and more robust. And then lastly, hopefully I've given you an idea of we can use multiplexing, and I think those types of automation techniques that have worked for larger scale chromatography can play a big role for bringing proteomics and nanospray to the clinical labs. So finally, I'll just conclude with a few acknowledgments. Clearly, I've had a great time working with Gary Velaskovic and his outstanding team at New Objective. Some of this early work was done while I was still at Merck, and Katie Southwick was the mass spectrometrist who really worked on making the picoslide work the first time we came up with the concept. She unfortunately didn't have the valve, so she had 56 connections to troubleshoot, so we were glad to get rid of that. And then when we moved to the University of Pittsburgh, Steve Mullett has picked this up along with Nick Stewart, and can't thank Valco enough. Their valves have made the technology really work well. So thank you. I'll take any questions.
Some questions?
Yes, please.
First, thank you for this very understandable talk. I wanted to know what you think about, so you told us about the depletion of proteins and affinity. So what do you think about the loss of protein species which do not bind due to post-translational modification? Would that have the result of the outcome of the diagnostic result?
So I just want to make sure I understand. So are you talking about immunodepletion to get rid of the most abundant proteins in plasma? Or so in the case of SDF1 alpha and GLP-1, neither of those is known to be phosphorylated that I'm aware of. And so we weren't worried about post-translational modifications. In that case, it was we knew exactly what form of those peptides we wanted to measure. But I think there are concerns that there's methods for cleaning up plasma and also for spinal fluid where you are doing immunodepletion to get rid of 10 or 20 of the most abundant proteins in plasma. And there you do have to worry about what those are taking with them. Are you losing the isoforms you're interested in, or any other types of modified peptides and proteins? It also makes things like calibration and putting in internal standards a lot more complicated. So in general, I would think for where we are today, avoiding the immunodepletion methods for building clinical assays would be what you'd want to do first. But overall, the problem of digesting proteins to peptides in immunodepleting plasma is currently remains unsolved. So it's a big area where we need some more work. Other questions? Great. Rick?
So, how about a pico chip with 3 columns inside instead of having 3 pico chips?
You could do that. You could have 1 3-column. There's some funny things that come up with this column is that the first time we set it up, we let it run over the weekend, and for one reason or another, one of our samples clogged one of the 2 columns. And normally if that happens on a single sprayer, you'd come in on Monday and you'd have absolutely no data. But with this system, you come in and you have 2/3 of your data. So it's kind of a nice advantage. I don't think, we also are spraying out of each column simultaneously, so getting the 2 other columns further away from the mass spectrometer. And I didn't show the slides, but one of the problems that makes electrospray or nano-electrospray irreproducible is particles depositing on the tip. And so by separating the 2 tips away, you have the opportunity to wash and clean off those tips while they're not being used in front of the mass spectrometer. But we were just adapting a single sprayer to a triple sprayer system. There's some applications. All right. Thank you very much.